Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Osmolality as a Tool for UF-DF Monitoring Before Cation Exchange Chromatography Capture of Monoclonal Antibodies. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Advanced Instruments who developed the content for this presentation. Advanced Instruments is a global company offering a novel portfolio of analytical tools, including Osmotech, a robust line of microosmometers to support bioprocessing and quality control, and Solentum, a portfolio of best-in-class imaging and single-cell disposition technologies for cell line development workflows and assurance of clonality for regulatory bodies. Now it's my pleasure to introduce you to your speaker for today's webinar, and he is Tiago Millen. He is the lead scientist at Jefferson Institute for Bioprocessing with a broad background in downstream process development. Since 2008, he has worked at fast-paced biotech startups and in a large pharmaceutical company. He has experience in optimizing protein expression and developing multi-step purification strategies for antibodies and other therapeutic proteins, and performing protein analytics to support process characterization studies. Now, before I pass over the controls to Tiago, we have our first poll question for our audience members to interact with in real time, and that's going to appear on your screen right now. And there it is. You can go ahead and vote on this question by clicking on any of the answers. And the question that we have for you today is, how many of you currently measure osmolality in your downstream processes? I'll give you about 30 seconds to complete this simple poll question, and your options are below. All the time, some of the time, none of the time, I wasn't aware osmolality could be used in downstream processes. So please go ahead and cast your vote. I appreciate that. I'll give a few more seconds, as it looks like there's a couple of you still there working on that poll question. So a few more seconds for you to launch it and to cast your vote. Perfect. That looks like it's the majority of our audience members. So thank you very much, everyone, for that. Let's take a quick look at the results. Okay, so here's the results of the first poll question. So the question was, how many of you currently measure osmolality in your downstream processes? And we got 21% are saying all the time, 29% are saying some of the time, then we have another 29% saying none of the time, and then we have 21% saying, I wasn't aware osmolality could be used in downstream. So thank you everyone for participating in our first poll question. Now it's my pleasure to pass over the mic and the controls over to Tiago. So Tiago, when you're ready, you may begin.
Thank you very much for the introduction, Sonia. Hello everyone, welcome to my presentation entitled Osmolality as a Tool for Ultrafiltration Diafiltration Monitoring Before Cation Exchange Chromatography Capture of Monoclonal Antibodies. I will start talking about the monoclonal antibodies manufacturing. The first monoclonal antibodies were approved by FDA in the late 90s, and since then, process has been optimized to the current platforms established today. I want to share a typical monoclonal antibody platform manufacturing process from the working cell bank ampoule to the drug product vial at the bottom. So the upstream operation starts with the seed train expansion and inoculation train in single-use bioreactors. After 2 to 3 weeks, the production bioreactor can be inoculated, and then after another 15 days on average, will be harvested to separate suspended impurities and provide cell-free material for the downstream operations. In my example, considering a production bioreactor up to 2,000 liters, the primary recovery will be performed by a series of depth filters, typically 3, plus a bioburden 0.22 micron filter. In the downstream unit operations, dissolved impurities like the host cell protein, host cell DNA, viruses will be removed. A 3-column process is the industry standard for the MABS purification, plus a comprehensive virus clearance strategy, here described as detergent viral activation and low pH hold, and a nanofiltration operation. The typical capture column is packed with protein A resin, which has affinity for the Fc portion of monoclonal antibodies, and the elution is achieved in low pH. Following, the low pH hold is the natural subsequent operation, and it can be coupled to host cell protein precipitation. Then, the product conductivity MPa is adjusted to be loaded onto the second purification column, like a cation exchange in binding elute mode. The cation exchange eluate needs to be adjusted before loading onto the polishing chromatograph column or membrane, which for most molecules is an anion exchange column operated in flow-through mode, given the isoelectric point of the majority of the MABs, so they will not bind to an AEX resin, only the impurities carrying a negative net charge. Finally, the nanofiltration is performed to complete the viral clearance, and the product is concentrated by tangential flow filtration, followed by the ultrafiltration diafiltration operation to buffer exchange into a proper buffer to preserve the molecule stability during the API freeze-thaw operation and further drug product formulation. I want to bring some protein A benchmark. By the time the first MABs were approved, protein A resin was not available. When they were commercially available in the early 2000s, companies worried about continuous resin supply. The protein A chromatography is currently the industry standard capture column in the monoclonal antibody platform process due to its high selectivity and robustness. Protein A is typically insensitive to feed conditions, and recent advances in the surface chemistry, engineered protein A, have improved binding capacities and reduced leached protein A. At the bottom, we can see protein A in the light blue arrow binding to a monoclonal antibody in the Fc portion, the dark blue molecule. On the right, you can see the Fc portion highlighted in purple. However, as the industry moves to higher upstream titers, over 10 grams per liter, and higher cell densities, over 80 million cells per ml, the demands on the capture column are increasing. So the increased titer requires either larger columns or more cycles per batch of culture fluid, which has a dramatic impact on processing times and cost. And also, current supply chain delays may put on risk platform process based on the protein A resin capture. Next, I will show how we can apply osmolality as a tool for bioprocess monitoring. What is osmolality? It is a measure of total solute concentration. A solution is the solvent plus the solute, and osmolality will not measure undissolved particles. It will measure dissociated ions. And why does it matter in the upstream operation? So it is a good indicator of media performance. So most mammalian cells can tolerate a range of 260 to 320 milliosmoles per kilo, on average 290 milliosmoles per kilo plus minus 10%. It affects the osmotic flow, the cell size, intracellular concentration, as you can see in the example of isotonic media, hypotonic or hypertonic media, causing cell shrinking. Also, osmolality is an excellent quality control measurement to determine if all correct components were added in the correct amounts. It also helps in demonstrating the precision of a manufacturing process for weighing, dissolving, filtering, and preparation of materials. Osmolality has been used as a standard measurement of solute concentration in biopharma for decades. We used the Osmotec XT single-use micro-osmometer for the entirety of this study, and we found the osmometer was easy to use and highly accurate and was well suited for all aspects of the study I'm presenting on today. It is designed for biotech applications, being the only osmometer with widest range of osmolality testing and performance for the different types of samples seen across bioprocessing with freezing point technology. So the main features are accurate and precise osmolality, results across the largest range from 0 to 4,000 milliosmolar per kilo, ability to freeze complex sample types across bioprocessing workflow, quick and user-friendly, complete traceability and efficient management of electronic data, comprehensive 21 CFR Part 11 compliant features such as electronic signature capabilities. Typically, osmolality is used for upstream monitor, as you can see in this CHO cell culture expressing a monoclonal antibody in fed batch mode. In this example, the initial media osmolality on day 0 was 363 milliosmolar per kilo, and the osmolality was monitored over 12 days of culture and increased as the feed strategy was applied, as described. Another application for osmolality is in downstream operations. Conventional cation exchange resin have low selectivity for antibodies in clarified cell culture media under typical media conductivity. Therefore, a CX capture column requires dilution of the feed or concentration and diafiltration step to adjust the pH to the 5 to 6 range prior to loading to achieve a reasonable bind to the column. In a simplified downstream unit operations proposed here, the osmolality monitoring can be useful during the tangential flow filtration, the ultrafiltration diafiltration conditioning, before loading the CX capture column, and during the purification. It is also important parameter to monitor during the final product concentration and buffer exchange into the product formulation buffer. Next, I will show the development of a downstream purification strategy using a cation exchange resin as the capture column. To conduct the evaluations for this project, Jefferson Institute for Bioprocessing used a CHO cell line expressing a monoclonal antibody, rituximab, in a specified growth medium. So this platform cell line and medium were used to create cell-free material for the downstream studies. A single-use 20-liter wave bioreactor from Cytiva with a 10-liter working volume was used for production. So the bioreactor was seated at a density of 0.3 million cells per ml in the balanced D media. During the course of the experiment, the cell culture was operated in fed batch mode, and samples were taken and analyzed daily to assess the cell growth, viability, and metabolites profile. So the cell-free material was then passed through a 0.22 micron filter, and the CFM turbidity was 15 NTU after filter. So after harvesting, the cell-free media conductivity was 12.9 millisiemens per centimeter and pH 7.2, with osmolality of 403 milliosmolar per kilo. So the cell-free media was concentrated by TFF and buffer exchanged by UFDF into an appropriate buffer to allow binding to the cation exchange resin. So the conductivity and pH adjustment are crucial for the CX capture step. A prepacked high-trap capital S impact 1 ml column from Cytiva was used as the scale-down model during the process development phase. So the binding, wash, and elution buffers were optimized to maximize the capture yield and the protein purity. I will give a brief explanation about the operation of TFF, where the majority of the flow is direct across or tangential to the filter membrane, filtration model, or a hollow fiber model. A fraction of the flow that's permeating through the membrane is called a permeate flow. The system consists in a feed tank connected to a pump that feeds the TFF model. The permeate stream is collected and the retentate stream, or concentrate, returns to the feed tank. Pressure sensors monitor the feed, retentate, and permeate pressure, allow us to calculate the transmembrane pressure, TMP, which is kept constant along the operation. The feed volume will reduce over time as we collect more and more permeate stream. Then the initial feed will be concentrated. For the UFDF operation, we will start with a defined volume of feed, which is called diavolume, and then fresh buffer can be added onto the feed tank at the same rate the permeate stream is collected, resulting in a complete buffer exchange after the determined volume of diafiltration, fresh buffer, being added on the top of the original feed solution. So typically 7 to 8 diavolumes are necessary to achieve a complete buffer exchange. After the upstream operation harvest, we started with 2.6 liter of selfie media. A threefold concentration was performed using a 50 square centimeter Pelican XL TFF cassette with a 50 kilodalton molecular weight cutoff from millipore. So the flow rate was 35 mL per minute and the transmembrane pressure TMP was set at 22 psi. So during the buffer exchange, the permeate flux decay was severe, especially when the pH went close to 6.0. The cloudiness was intense. A lot of host cell proteins start to precipitate. So here I highlighted the critical pH range where we observed the increased cloudiness. At the end of the concentration, the material was clarified to remove cloudiness, filtering through a 0.22 micron membrane filter. And the clarified material was buffer exchange in a total of 4 diavolumes, and the retentate or concentrate and permeate were monitored as shown below. For the preparative purification, we use the same S impact resin from Cytiva, which has an average particle diameter of 50 micron. So these are the buffers used. So for the column equilibration, 25 millimolar sodium acetate, 50 millimolar sodium chloride at pH 5 with a conductivity of 6.44 millisiemens per centimeter and osmolality of 133 milliosm per kilo. So we kept the same sodium acetate concentration and pH for the other buffers. So the wash buffer has 100 millimolar sodium chloride with a conductivity of 10.9 millisiemens per centimeter and osmolality of 218 milliosm per kilo. And the elution buffer was 500 millimolar sodium chloride with a conductivity at 46 millisiemens per centimeter and osmolality 939 milliosm per kilo. Then a 10 centimeter bed height column was packed in a 1.6 centimeter diameter column body with a total resin volume of 20.7 mL, and the column efficiency was assessed by HATP and asymmetry tests. So the concentrated and conditioning CFM was loaded onto the column with a 3-minute residence time, and the wash step was performed with 8 column volumes, and the elution over a 20 column volume gradient ranging from 10 to 65% of the elution buffer. Here we have the chromatogram showing the sample load, wash step, and elution over a 20 column volume gradient. So the UV at 280 nanometers is shown as the blue trace, and the inline conductivity monitoring is the orange trace. So we monitor the osmolality, conductivity, and pH of all steps as shown on this table. So it includes the load, the flow-through fraction, the wash, elution, and strip. So all osmolality readings were accordingly to the expected as confirmed by the conductivity readings. So the expected osmolality for the load sample to allow proper binding to the CX resin is between 160 and 175 milliosm per kilo, which represents a conductivity around 7.5 millisiemens per centimeter. We measured the titer pre and post purification, and the titer was assessed by analytical HPLC column, a MapPack Pro Protein A column from Thermo Scientific, and we prepare a mass balance for the CX capture step. So the CX column yield was 92%, which is consistent with the expectation for a capture step based on affinity chromatography, typically over 90%. So samples were analyzed by SDS page using a 4 to 12% gradient gel to assess the purity. On lane 1, we have a molecular weight ladder ranging from 185 to 10 kilodalton. On lane 2, we have an IgG standard in non-reduced, showing an expected molecular weight of 150 kilodalton. So lane 3, we have a self-feed media after harvesting. On lane 4, the capital S load after the UFDF conditioning and buffer exchange. So on lane 5, we have our column flow through, showing the molecule of interest actually bound to the column. On lane 6, we have the column wash, where we can see some impurities removed. Lane 7, we have our capital S solution. First, the non-reduced, where we can see the intact monoclonal antibody with the expected mass around 150 kilodalton. On lane 8, we have our capital S solution under reduced condition, which is consistent with the expected molecular weight for the heavy chain, around 50 kilodalton, and the light chain, around 25 kilodalton. And last, we have our column strip with the remaining impurities in the column. Next, we performed a polishing step with an ion exchange resin to remove process-related impurities, such as host cell DNA, host cell protein, viruses, and endotoxins. So the capital S eluate material was filtered with a 0.22 micron membrane filter, and due to the high conductivity after the CX solution, which was 22.8 millisiemens per centimeter, a sample was fourfold diluted into the AX equilibration buffer in order to bring down the conductivity. So the AX equilibration buffer is a 10 millimolar sodium phosphate, pH 7.3, with a conductivity of 3.35 millisiemens per centimeter and osmolality of 47 milliosmol per kilo. So the buffer pH was chosen based on the molecule isoelectric point, which is 8.3. So the Q-Cephalose fast flow resin from Cytiva with an average particle diameter of 90 micron was packed to achieve a 5 centimeter bed height column in a 1.6 centimeter column body. So the column efficiency was assessed by the HTTP and asymmetry, and both passed. So the column was operated in flow-through mode, where the contaminants will bind to the column and the protein of interest is collected in the flow-through fraction. So the sample was loaded with a 2-minute residence time, and the flow-through and the column wash, total of 3 column volume, were collected together to minimize the product losses. So the sample load conditions are described. Osmolality was 179 milliosmol per kilo, conductivity 10.1 millisiemens per centimeter, and pH 7.3. So the wash buffer has the same composition as the equilibration buffer, 10 millimolar sodium phosphate at pH 7.3. And we used the osmolality readings for this step as quality control for the buffers and load condition. So the optimal range for osmolality for this AEX process operating in a flow-through mode is between 170 to 180 milliosmol per kilo, and the optimal range in conductivity for this step is from 8 to 10 millisiemens per centimeter. So the titer was assessed by analytical protein A HPLC to prepare the AEX mass balance that show a step yield of 97%. So the protein concentration after capital S solution was 3.3 mg per ml, and after a fourfold dilution, the Q-Cephalose load concentration was 0.82 mg per ml, and the collective fraction, the flow-through plus the wash, was 0.75 mg per ml. So that gives us a step yield of 90%, which is considered high for this type of operation. After the AEX column, the final product concentration and buffer exchange was performed. So first, the material was concentrated 26-fold using a 50 square centimeter Pellicon XL TFF cassette with a 50 kilodalton molecular weight cutoff membrane from millipore. So the flow rate was 35 mL per minute and the transmembrane pressure was set at 22 psi. So the resultant volume was approximately 20 mL with a MAP concentration of approximately 20 mg per ml. So this material was buffer exchange into rituximab formulation buffer, which is 7.35 mg per ml of sodium citrate, 9 mg per ml of sodium chloride, 0.7 mg per ml of polysorbate 8 at pH 6.5. Conductivity for the final formulation buffer is 23 millisiemens per centimeter, and the osmolality was 403 milliosmolar per kilo. So the concentrate and the permeate were more, as shown in the table below. So for the UFDF run, one diavolume was equivalent to 20 ml, and the buffer exchange was completed when 8 diavolumes were achieved and the final product matched the same parameters of the formulation buffer. We can see the formulation buffer specs at the bottom line in the table, and after achieving 8 diavolumes, we almost matched that specification. Next, I will present our findings after analyzing the samples collected. Just to remember the unit operations performed, so we start harvesting the cell culture material from the bioreactor to obtain a cell-free material, and then tangential flow filtration and UFDF was performed to condition the samples before loading the CX column. So after the CX capture step was performed using the capital S resin, and the next operation was a polishing step using an anion exchange resin, Q-Cephalose, FF, and following TFF operation to concentrate and UFDF for the buffer exchange into the final formulation buffer for this monoclonal antibody was performed. So the arrows highlight the steps where we collected samples and evaluate the osmolality. So we start to develop a model based on the UFDF operations described here. Based on the osmolality and conductivity data from samples during the downstream process, we started to develop a model to understand the utility of near-line monitoring of osmolality of cell-free media as a function of impact on the CX capture. So from the concentrate samples data, we plotted a curve on jump, correlated the conductivity and osmolality changes along the UFDS process in diavolumes on the x-axis to condition the cell-free media for loading onto the CX column. So in red, we have the osmolality data, and in blue, our conductivity data. So the first plot is the relative data. So we consider our initial condition as 100%, and then after the buffer exchange, after 4 diavolumes, we achieved a percentage reduction in the initial conductivity and osmolality values. So on the left Y, we see the osmolality decrease. On the right Y-axis, we see the conductivity decreases. So we also normalized the data points, adjusting by a logarithmic fit based on the total changes of conductivity on osmolality, ranging from 1.0, the initial values, to 0, the final values, shown on the right panel. So the normalized data is on the right, so adjusted by the same logarithmic fit. And red circles are osmolality and blue crosses are the conductivity. So you can see the initial conditions starting at 1.0, and over the 4 diavolumes performed, you can see the decrease it close to the zero, or is the final buffer condition. Here we have the permeate samples data collected during the buffer exchange to condition sample before loading the CEX column. So the relative osmolality data is in red, and the conductivity data is in blue on the left panels. We also prepare normalized data points adjusted by a smooth linear fit in the right panel. So we can observe the same good correlation between the change in the osmolality when compared to the change in the conductivity. So confirming that osmolality is a good tool to monitor the change during the UFDF operations for this type of cell-free media conditioning before loading CEX capture column. Then we did the same analysis for the concentrated samples collected during the final product concentration and buffer exchange. We plotted a curve on jump correlating the conductivity and osmolality change along the UFDF process in diavolumes on the x-axis to achieve the complete buffer exchange into the final formulation buffer. So the relative osmolality data is in red and the conductivity data is in blue on that chart. So on the number of diavolumes can be observed on the x, and our initial conductivity and osmolality was lower than the final conductivity and osmolality of the formulation buffer. That's the reason why this curve is going up. So we also normalized the data points adjusted by a log fit. So based on the total change of conductivity and osmolality, again ranging from 1.0, the initial values, to 0, the final values, shown on the right panel. We can observe a good relationship between the osmolality and the conductivity data fed by the same equation. Repeating the same analysis for permeate samples collected during the final product concentration and buffer exchange. We have the relative data. On the left Y, we can see the osmolality change. The red line is the osmolality data, and the blue line is the conductivity data. On the right Y, we can see the conductivity change over the 8 diavolumes performed to complete the buffer exchange. So again, the curve is going up because the initial condition, conductivity and osmolality for the buffer are lower than the final formulation buffer. We also normalized the data points adjusted by log fit. So based on the total change of the conductivity and osmolality, again ranging from 1 initial values to 0, the final values, shown on the right graph. So again, we can see a good relationship between the conductivity and osmolality data for that type of UFDF operation. For the discussion and future work, I want to highlight our findings. So we observed that using a CEX column to capture rituximab was feasible after slight process adjustments. So the CEX capture and AEX polishing steps showed high yields and high level of protein purities according to our analysis. After the 2-column purification process, the final product concentration and buffer exchange into the formulation buffer was performed, achieving a rituximab concentration of 20 mg/ml. The osmolality monitoring performed during the UFDF operations, pre and post purification, showed a good correlation with the conductivity data as observed on the fed normalized data plots. It suggests that online osmolality monitoring might be used to control the UFDF process performance. Our findings indicate a high sensitivity of osmolality readings to the TFF and UFTF operation conditions. It might be a more powerful tool than conductivity to identify slight process deviations. Like if your initial buffer condition and the final buffer condition, like for the formulation buffer, they have close values for the conductivity. I believe the osmolality can bring more light to the slight change in that process. Like conductivity just change 1 to 2 millisiemens between the initial and the final buffer condition. I think the osmolality change, it's a broader range, so you can see very slight difference and follow the process a little bit better than monitoring by conductivity only. But the main challenge is to replace this traditionally established conductivity monitoring by osmolality monitoring. I believe introducing the osmolality monitoring in new downstream process, such as viral vector purification, would be interesting. Viral vector purification still lacks a robust platform process. To achieve that, high throughput osmolality measurement system would be helpful for process development and process characterization studies before establishing a robust manufacturing platform. I would like to thank Advanced Instruments for the opportunity of collaboration and development of this project. Thanks to Mark Wardenberg, Advanced Instruments Company Liaison. Thanks for the data discussion. Also, thanks for Cameron, our Director of Process Development and Operations, and to TIB staff for the analytical support and providing the cell culture material, and also for the data discussion. Thank you to the audience. Pleasure to answer your questions about the results presented, and Advanced Instruments team may help with questions regarding the equipment used. Sonia will manage the question and answer portion. Thank you again.
Well, thank you very much, Tiago, for that very in-depth presentation. I hope everyone enjoyed it. And before we start the Q&A portion of the webinar, we have one last poll question for our audience members to engage in. I'm going to launch it now. And just like before, it's done in real time, so your participation is strongly encouraged and very much appreciated. And all you have to do is click on any of the answers below. Okay, and the question that we have here is, after listening to this presentation, would you consider using osmolality in your downstream process? We have a couple of options for you to choose from. Yes, no, unsure, or we would need more data to show proof of principle. So please go ahead and cast your votes. And then once I close the polls, we'll get right into the Q&A. So remember to send your questions through using that questions window. Okay, so it looks like we got the majority of our audience have cast their votes. So thank you very much for that. Just going to give a few more seconds. Okay, that looks like it's the majority of everyone. All right, I'm now going to close the polls and share the results. Okay, so here are the results. We have here 37% of our audience members are saying yes, 0% are saying no, 47% are saying unsure, and then we have 16% said that we would need more data to show proof of principle. So thank you everyone for participating in our final poll question. And now we're going to launch into the Q&A portion of the webinar. And with that, we'll put on our webcams. And I'll invite Tiago to put on his webcam. Here I am. Hi, everyone. And there's Tiago. Thank you. All right. So did you want to comment on the results of that last poll question?
Yes, I'm impressed with 50% of the audience applying the osmolality measure in the downstream. So let's work for the next, for the other 50%, start to be using it.
Okay, perfect. Thank you. All right. So remember, audience members, to send in your questions by using that questions window that's there on the right-hand side of your screen, and we'll try to get your questions in during this time that we have together. I have some questions that have already come in, so I'm going to start with the first one here. So this audience member is asking, why do you not use Pro-A as a capture step?
Well, that's the standard in the industry. We just look for an alternative. So CX has been proven as an appropriate alternative for the capture step for monoclonal antibodies. And we just want to highlight the importance of monitoring of osmolality, as well the conductivity for the binding conditions onto the CX resin, and bring the importance of the osmolality monitor, especially for the ultrafiltration diafiltration conditioning.
Okay, all right, thank you. Here's our next question. This audience member is saying, when would osmo and con mismatch be a problem? Was that a concern for this process train at all? It is not clear to me what utility this data set is providing at this stage.
For this process train, I think the conductivity and osmolality readings, they match the expected values for the buffers prepared, for the protein condition before loading the columns. And I think it helps us, especially the classical instructions for ultrafiltration diafiltration is to perform 7 to 8 diavolumes to achieve a complete buffer exchange. But for this process conditioning for the capture on the CX column, we notice just performing 4 diavolumes allow us to make the sample ready for a proper binding onto the CX resin. So that saves us a lot of time and resource, especially in the lab for these large volumes could be an issue to process 8 diavolumes before starting the capture and completing all the steps on the same day in the lab.
Okay, thank you, Tiago. All right, here's the next question. This audience member is asking, in your presentation, you mentioned that osmolality is important for media performance and as a QC measurement. Can you go into more detail on where you think osmolality fits in the downstream process?
Well, osmolality can also be used as a quality control measurement in the downstream. Buffer preparation and feed conditioning are crucial for proper column binding. And also these UFDF operations can also be monitored by osmolality.
Okay, let's go on to the next question here. As you work through your process development, where do you think osmolality holds the greatest advantage over conductivity?
Well, I think the main advantage of osmolality over conductivity is when the initial and final conditions have similar readings for the conductivity, like 1 or 2 millisiemens of difference between the initial and final buffer condition. And then in that case, the measurement of slight change in the process may be challenging. I think osmolality can help monitoring these very, very discrete changes in the buffer condition.
Okay, thank you. Let's go on to our next question. Just a reminder to the audience members, so please send your questions through using that questions window. This looks like it may be our last question, but if you have any questions, please go ahead and utilize that questions window. All right, so this audience member is asking, based on your work, would you hypothesize that using osmolality as a tool could improve the overall downstream filtration workflow?
Yeah, buffer exchange operations may be necessary between chromatograph columns in order to enable the mainstream sample to bind to the next chromatograph resin. And to achieve that, close osmolality monitoring may help to minimize the number of diavolumes needed. So we can limit the operation to the point the mainstream matches the binding buffer condition, and that will save us time and resources.
Sorry about that. I was just trying to get these lined up here. Okay. All right. So that looks like it's it for our questions. So thank you very much, everyone, for sending those questions in. It looks like we have reached the end of the Q&A portion of the webinar. If we couldn't attend to your questions, Tiago, or you can reach out to Mark R at aicompanies.com. They'd be more than happy to further give you details regarding your questions or any of your inquiries. Okay, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, give me a few seconds. I'm going to send you a link in your chat box and you'll be able to view the recording of this event with that link and also share that link with your colleagues once they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speaker, Tiago, for that very insightful presentation and for answering your questions. So thank you, Tiago.
Thank you very much.
Okay, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Thank you again, Tiago. Thank you, everyone.
Bye.
Bye.